Jai Hind students, let us learn the next subheading Linkage and Recombination under chapter 5. Linkage. Linkage refers to the physical association of genes that are present on a chromosome. For example, human has 23 linkage groups. Drosophila has 4 linkage groups. These are always haploid. Recombination. The rearrangement of linked genes due to crossing over is known as recombination. The genes present on the same chromosomes tend to be inherited together are called linked genes, and this phenomenon is called linkage. The recombinant frequency of linked genes is always less than 50%. All the linked genes of a chromosome form a linkage group. The number of linkage groups correspond to the haploid number of chromosomes. For example, there are 4 linkage groups in Drosophila, 23 in human. Morgan carried out several dihybrid crosses in Drosophila with the genes that were sex-linked. Example, the genes that are present in X chromosome. He hybridized yellow-bodied, white-eyed females to brown-bodied, red-eyed males and intercrossed their F1 progeny. He observed that the 2 genes did not segregate independently of each other, and the F2 ratio deviated very significantly from the 9:3:3:1 ratio. He concluded when the 2 genes in a dihybrid cross are situated on the same chromosome, the proportion of parental gene combinations are much higher than the non-parental type. Morgan explained these results were due to physical association, that is linkage of the 2 genes. He coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the formation of non-parental gene combinations, that is new combination. Morgan found that some genes present on the same chromosome were very tightly linked, showed very low recombination, where some others loosely linked showed higher recombinations. They observed that white and yellow genes were tightly linked with 1.3% recombination, and white-eyed and miniature wing usually showed 37.2% of recombination. Alfred Stuyvesant determined that genes of Drosophila are arranged in a linear order. He measured the distance between genes and prepared chromosome maps with the position of genes on the chromosomes based on percentage of recombinants. These are also called genetic maps. Gene frequency is equal to total number of recombinants divided by total number of progeny. Genetic map. The assessment of relative position of genes on the chromosome produces the genetic map of that chromosome. Morgan unit. The frequency of 1% crossing over between 2 genes is taken to represent Morgan. Map unit. Map unit refers to a measure of genetic distance corresponding to a recombination frequency of 1% centimorgan. Centimorgan. Recombination frequency of 0.01% is equivalent to 1 map unit distance and is referred as centimorgan. See